I know. You left it like 1:30.
All right, everyone. We have a lot of material as well as a journal club to go over today. So my hope is to try and cover as much material as possible. Whatever we don't cover, I will try and record an addendum video and post onto Canvas. But again, I'm gonna try and make sure we cover as much as we can while not making the mistake of last journal club where I think I rushed through like the last 5 to 10 slides in like a heartbeat and expected you guys to memorize everything. So I'm trying to make sure we cover as much as we can while not trying to fly through it. So the last unit was really focusing on central dogma of how DNA begets more DNA through replication, but through transcription, we go from DNA to RNA, and through translation, we can go from RNA to protein. There's a fair number of elaborations around central dogma that that's gonna be the focus of our lecture today. A lot of it is both a combination of can be fairly complicated as well as still active areas of research. And so trying to hold those two things together, we're going to try and focus on what is known while trying to underline and make sure you guys understand like what is not yet known or not characterized, but is still active areas of research. We'll be looking at these elaborations on central dogma. We can also call it extradogma through the lens of two syndromes, Klinefelter syndrome and Down syndrome. Down syndrome is a genetic disorder with a wide variety of both developmental delays as well as physical presentations. A lot of us probably have either close friends or family or people we've known growing up who have Down syndrome. But some of the key things to note about Down syndrome is that there's some of the physical presentations of it are brachycephaly, which is a shorter head, brachydactyly, which is typically shorter digits. There's also a duodenal atresia. Atresia is usually either a shortening or a closing off or obstruction of certain cavities in the body. So this is an atresia of the duodenum of the GI tract. So there's usually either a closing or an obstruction of the duodenum. There's also epicanthal folds. It's these the folding over of their top eyelid over the corner. That's usually another physical presentation. There's also fifth finger clinodactyly, which is usually a curving or bending of the fifth digit. There's hypotonia, low muscle tone, and as a result of that low muscle tone, there's also lax ligaments. There's the intellectual manifestations of Down syndrome, but in addition to that, there's also general short stature and an effect on their toe gap as well. The key thing to note is that Down syndrome is it's a genetic disorder by nature of having a third copy of the 21st chromosome. Another chromosomal disorder that we're going to focus on is called Klinefelter syndrome. It's the most common genetic cause of male infertility. It is because of sometimes how difficult it can be to try and diagnose. Oftentimes, as many as 65% of cases can go undiagnosed. Some of the ways in which it manifests is gynecomastia, which is development of breast tissue in male patients. There's hypogonadism, which is the underdevelopment of sex organs. And finally, there's the elevated levels of follicle-stimulating hormone. There's this basically a good way to try and kind of think about it or characterize it overall is there's these large hormonal shifts that present in patients with Klinefelter syndrome. And a key thing to note is that here it's a extra copy of one of that sex chromosomes. As far as the numbers go, they're pretty close together in terms of both in prevalence and incidence, both at about 250,000 patients in the US, as well as at about 1 to 500 or 1 to 1,000 births in the US. What's important to know is that it appears to occur randomly. There's no known behavioral or environmental factor that increases the likelihood. The only factor that has been associated with either of these is that it's been identified that a contributing factor for Down syndrome is that as the age of the mother increases, the likelihood of Down syndrome occurring goes up. Again, it's not causal, meaning that if this happens, this definitely happens, but rather there's a reported relationship between the two. as for etiology, both of these are caused by a defective meiosis and then aneuploidy. So meiosis, so we've talked about mitosis, that is cell replication of somatic cells within our body. We've talked about fibroblasts, we've talked about immune cells. The cell replication of those cells is called mitosis. Meiosis is cell division of gametes, of sperms, of germ cells, primarily here of sperm and egg cells, or we can just refer to them as gametes. So mitosis is primarily for somatic cells. Meiosis is they're both cell division, but meiosis was primarily for gametes here. And then aneuploidy is a term that means it is characterized, it means extra copies of chromosomes. So both Down syndrome and Klinefelter are diseases of aneuploidy or disorders of aneuploidy. So Down syndrome is one extra chromosome of chromosome 21. Because we typically have 2 copies, that means now in Down syndrome there's 3 copies. We can also call this trisomy 21. Whenever you hear trisomy, that typically means 3 copies of whatever that chromosome is. In Klinefelter syndrome, we don't typically use the term trisomy here because typically in men there's 1 X and 1 Y chromosome, and in women there's 2 X chromosomes. So typically here in Klinefelter syndrome, there is an extra X chromosome. There's something to consider here where in chromosomal aneuploidy, where we have these extra copies of chromosomes, there's a gene dosage issue, meaning if there's an extra chromosome, that means there's more DNA that can be transcribed that leads to more protein that's encoded for on that chromosome. But what's important to note, or a good question for us to think about is, it's clear that there are different degrees of the symptoms that present with both Down syndrome and Klinefelter syndrome are both different in severity. But if they're both cases of aneuploidy, why are their symptoms so stark and so different? One thought might be that perhaps chromosome 21 and the X chromosome are really different in size. It cannot be explained by that because chromosome 21 is actually a lot smaller. It's about 48 megabases long, which has about 250 genes on it, whereas the X chromosome is a lot larger, has about 150 megabases on it, and equates to about 150 genes. So the size, so it can be explained by chromosome 21 being a lot larger here. So what we're going to do is we're going to be able to answer this question, hopefully get to be able to answer this question at the end of the lecture before we have to start our journal club. So keep these questions in your mind before we get back to it. So there are elaborations that we're going to focus on today. The first elaboration on central dogma is going to be something called epigenetics. Epi means on or around. So that means these are going to be changes that are on or around the DNA. We're not making changes. We are not making changes to the DNA sequences, but rather making modifications on or around the DNA. There's a few definitions of epigenetics that have kind of emerged over the years. There's one that's a little more traditional. The idea is that these epigenetic changes, or these changes on or around the DNA, are stable and heritable modifications that impact the genome, our entire DNA, and its regulation of it, but they do not alter the actual sequences, the nucleotides themselves. So the key thing to note with the traditional definition is that these are stable, inheritable, and inheritable changes. A more recent definition is that epigenetics is the structural adaptation or changes of our chromosomes and regions along our chromosomes, so as to try and promote altered activity states where these are more transient, a little more temporary, and not inheritable or stable. So the distinction here is that one is more stable and inheritable and passed down to progeny, and one is more kind of like a state that we go in and out of. The reality is that it's both, that they are stable, heritable changes that we inherit from our parents, as well as some changes that occur that can change throughout the day or on the order of days. So example of stable and heritable epigenetics. One example is something that's called the agouti mouse. So it's this mouse that has a certain color, certain coat color. And what ends up happening is that based on changes in the mother's diet while she is pregnant, there are changes to the locus, or locus means like region of DNA that's responsible for this. The locus that regulates coat color of the progeny is affected by the mom's diet, and it leads to a permanent change in the coat color of the mice. So if we so if you look at the image here, you can see that the entire litter of mice who were born from a mom who had her diet changed that subsequently had an effect on the agouti locus, or the locus that regulates color of the fur. There was actually a stable heritable change there. And so a lot of this indicates that there's actual stable change that can occur. A few other changes that a few other things that also communicate this is that there are in instances of communities where either starvation or poor access to food can cause stable heritable changes that are passed down to progeny, where it can actually lead to metabolic changes that are passed down to generations. Another example to consider is that actually smoking can actually lead to stable heritable changes, where parents who smoke can actually lead to stable heritable changes where their progeny or their children can actually be more predisposed to becoming asthmatic. So those are examples of stable and heritable changes that are these are inherited, but they're not changing the actual DNA sequence here. Other examples that are more transient, plastic means you can go in and out of it, are circadian rhythm genes. These are genes where their regulation is affected. Again, the sequences are not being changed, but circadian rhythm can lead to changes in total gene expression of a variety of genes. This is an entire new area of research that is actually making a lot of scientists consider, like, when do we actually collect our samples? At what time point of the day should we collect our samples? And so there's a whole network of circadian clock genes that regulate this. Another that's more related to neuroscience is the whole idea of learning and memory. When we go through the process of learning, there are actual changes at the gene expression level that try and encode for that memory. But as you all know, when we learn something, we can sometimes forget it. And so there's a change in that neuronal circuit where there's not a change in the DNA sequence, but there's a change in the regulation of that gene circuit, whereby now it's no longer being active and we have now forgotten. I'm sure that's something that none of us are familiar with. You guys remember everything, especially those class. So there's two primary epigenetic mechanisms that we're going to focus on in this class that control chromatin accessibility, the packing and unpacking of our chromatin that enables transcription to occur. The first is DNA methylation. As the name implies, this is happening on the DNA. This is where methyl group is added onto our DNA, and this is actually inducing compaction. This is going to eventually, the outcome of this is that's going to reduce or suppress gene transcription locally. The key part of locally is that typically this methylation is happening on a specific gene. It might be on, we talked about core promoters. The promoter can be methylated for that one specific gene, and that might affect transcription of that gene. Another key mechanism for us to consider is modification to the actual histones, so that the histones are the actual proteins that our DNA is wrapped around that forms a nucleosome, that then it creates this almost like bead with a string around it, the string being the DNA and the bead being this octamer of histones, H2A, H2B, H3, H4, all together, and each subsequent nucleosome creates this bead on a string that makes up our chromatin. There are many different modifications that can happen. We call them post-translational because it's happening to the histone, and again, histones are proteins. Some of these changes induce chromatin compaction that reduces transcription. If you can't access the DNA, it can't be transcribed. Other modifications will open it up. If it's open, now the DNA can be accessed, transcription can occur, and the gene can be expressed. And so we can, based on this, you can almost think of it as like a gas and a brake, where one is activating gene expression, one is repressing it. The combination of possible modifications here are astronomical. So especially for histone modifications, I want you guys to keep the big picture in mind because it gets fairly complicated, but I will take moments to try and draw your attention to things of what I want you guys to try and walk away knowing. I'm going to stop for a couple questions. I think I've been going for quite a while, so I'm going to stop for a couple questions here. Yeah, so for the sake of the recording, the question is, are there differences in inheritability with these two different kinds of modifications? To my knowledge, most of the inheritable ones come at the histone modification, the DNA methylation level. Histone modifications, it's a mixed bag because histone modifications can be fairly transient, but some of them can be slightly more long lasting. So it's a mixed bag with histone modifications. With the DNA methylation, to my knowledge, that's where more inheritable changes occur. Yeah, thanks for the question. So DNA methylation and silencing. So this is one slide where I'm not going to ask you to memorize this entire process. I'm going to draw your attention to 3 key enzymes or 3 key actors that make this process possible. So typically DNA methylation is happening particularly on cytosine here. That's one of our nucleotides at the DNA level. And so there's an enzyme called DNA methyltransferase. That is the enzyme, as the name implies, that is actually transferring or making the modification of adding a methyl group to our cytosines here. So you go from cytosine to 5-methylcytosine. The actual molecule that is serving as the donor or the provider of that methyl group is S-adenosyl methionine, or called SAM. So that's the source of the methyl group. And the enzyme that is mediating that transfer is DNA methyltransferase. And so from there, we have a few key, from 5-methylcytosine, we have a few mechanisms that enable us to remove or get rid of that methyl group. One of the key enzymes here is TET, and it's a demethylase where it can help remove and initiate the steps to remove that methyl group, where through both TET and base excision repair, we can get back from 5-methylcytosine to an unmethylated regular cytosine molecule here. And so again, we talked about this before, but particularly the regions, it's not just cytosine, but it's particularly CPG-rich regions called CPG islands. That's typically going to be within the promoter of a gene. So a lot of times where you're going to see methylation occur is not exclusively in a promoter, but where it's going to have its greatest effect is at a gene promoter. Again, that's right where transcription factors bind. That'll bring in TF2D and RNA Pol2 that promotes transcription from there. So if we have lots of methylated CPG islands within the promoter of a gene, that's going to lead to repressed gene expression, repressed transcription. So these methylation events that occur, we just walked over it. We just talked through how it can be removed. There's a really unique mechanism by which at the germline level or at the germ cell level, methylation events can be totally erased and then imprinted on our germ cells that then are passed down to the progeny once they become a fertilized egg. This entire process is called genomic imprinting. It's a little complicated, but again, start with the big picture. This is an entire process whereby at the primordial germ cells, you can think of these as the stem cells or precursors to our gametes, the precursors to the sperm and egg, whereby there are methylation events that are present throughout. The pink being the precursor or stem cell for an egg or for an oocyte, or the blue being the precursor or stem cell for a sperm. But you can just think of these as precursors for gametes. What ends up happening is whatever methylation events that are there are totally erased. They are all gone. And then methylation events are passed down simply by nature of whether this is a sperm or egg. And that's how we end up inheriting certain methylation events on our DNA that are inherited from our parents. One set of methylation events from the mother and one set of methylation events from the father, simply by nature of being either a sperm or egg. And so what this leads to is we have about 25,000 genes. About 300 of them are regulated by this process where they undergo methylation events that are imprinted at development, and passed down from the previous generation. So what this looks like is we have IGF-2 and the IGF-2 receptor. IGF-2, the copy of it that we get is only expressed by some, and so what I should say is this genomic imprinting, these methylation events, what ends up happening is that we have 2 copies of every gene, but these methylation events at the DNA lead to repression of them. So we only get 1 actual copy from mom and 1 actual copy from dad because of these methylation events. So what that leads to is that there's only 1 copy of IGF-2 that we get that's passed down from the father. And it promotes typically IGF signaling, leads to offspring, the growth of the fertilized egg. And the IGF-2 receptor, that copy, we only get from mom. And so that receptor, the modification, the expression that we get from it is, and the outcome of its signaling leads to restraining offspring growth. It's a decoy receptor for IGF-2. It's a sink. So more IGF-2 signaling leads to the growth of the fertilized embryo. And IGF-2 receptor is a sink for IGF-2, and it inhibits its signaling and is restricting it. And so each copy, the IGF-2, we only get a copy from dad. The IGF-2 decoy receptor, we only get a copy from mom. But the key thing to take away is that there are methylation events that are erased during development and reestablished, and that lead to the formation of the sperm and egg having unique methylation events that are only found in the sperm or egg that then are passed down to the progeny. Are there any questions on this slide here? Let me rephrase that. I'm going to stop for 2 questions here. Yeah. The question is, why are those methylation events erased? So these methylation events are erased to make space for the methylation. So let me take a step back. Even at the primordial germ cell, which is like the precursor or stem cell for our gametes, for our sperm and eggs, they're still a cell. They still have 2 copies of every chromosome. And there are methylation events that are inherited from the previous generation of mom and dad. Now that it's going to try and make a new sperm or a new egg, it's going to erase all of those methylation events and now only make methylation events that correspond to either being a sperm or egg, divide, and now only have 1 copy of our chromosomes, because now gametes only have 1 copy of chromosomes, and now be ready to pass that down as a gamete to create a fertilized embryo. Does that help clarify? I appreciate that question because I actually touched on something I didn't fully explain. All right, we'll go ahead and keep pressing on. So the histone modifications that we're going to talk about, the key thing to note here is that they will actually dictate or determine the accessibility of our chromatin. They'll dictate the unpacking of our chromatin and how accessible they are. The modifications of DNA that we just talked about are exclusively going to, the DNA methylation is going to act on the DNA and regulate really local effects of suppression. So with the modifications we have at the histone level, There's a variety of modifications that we can talk about, but one example that we're going to describe here is that we can have acetylation of amino acids on our histones. So again, key thing to note here, histones are a protein. They're made up of amino acids. So these modifications are going to be happening at specific amino acids on specific histones here. And the nucleosome, again, it's made up of H2A, H2B, H3, and H4. And there's a unique set of enzymes that mediate either adding an acetyl group or removing it. HAT is a histone acetyltransferase. It's going to be adding acetyl groups there. And HDAC is histone deacetylase. It'll be removing acetyl groups here. And the nomenclature we use to try and describe what the modification is starts by first identifying what the histone is. Here it's H3. The next is that what is the amino acid at which this is happening. So here it's the ninth amino acid, which is a lysine. So that's why it says K9. And you can see that here, that it's the ninth amino acid in the sequence, which is a lysine. And it's adding an acetyl group. So H3K9 AC means an acetylation at lysine at position 9 on histone H3. And so there's lots of changes you can make that communicate what the modification is at the histone level. And we'll show a few of these in a slide or two. And so there's a variety of histone marks. The marks means modifications that function with different kinetics. They have different time scales, and they're going to have different outcomes. So again, I'm going to repeat what I said earlier. I am not asking you to memorize what is the effect of each and every single histone modification. It's wildly complicated. I get it mixed up myself. So I'm not asking you all to memorize what is the effect of certain histone modifications. What we're going to have is certain terms in blue. They're going to draw your attention to what you should note, and I'll make sure to draw your attention to that. So we have a few different modifications that can happen at the histone level. We just covered acetylation, and that typically happens on the order of minutes. Another modification that we've talked about before, but we're going to bring it back here to histone modifications is phosphorylations. Both phosphorylation and acetylation is usually associated with active transcription. It's not a hard and fast rule, but it's a typical association that you can make. This is happening on the order of minutes to hours. And so one modification, if you remember, there's 3 specific amino acids that can be phosphorylated, serine, threonine, and tyrosine. So here, an example of using the nomenclature using the shorthand for histone modifications, we have a phosphorylation event at a serine that's at position 10 on histone 3. So typically what I do is I'll read this shorthand from right to left. That's typically the easiest way to make sense of it. The final histone modification is a methylation. We can actually have methylation that happens. We talked about methylation on DNA. We can also have it happen on histones. What's different is that we typically talk about methylation with DNA as happening with a single methyl group. With histones, we can have multiple methyl groups being added here. So we can have a single methylation that's a monomethyl. This is all happening on a lysine. Monomethyl lysine, dimethyl lysine, or trimethyl. So we can have 1, 2, or 3 methyl groups added. And the enzymes that mediate the adding of methyl groups to our histones is histone methyltransferase. And the enzyme that can remove these methylation events are histone demethylase. And the combination of what can happen as a result of methylation varies based on what histone we're modifying, where along that histone we're modifying it, and how many of those methyl groups are being added. So for example, just looking at histone H3, if we look at the fourth position of lysine, if we're adding 2 or 3 methyl groups, that'll promote its activity. On the same histone, if we go over to the 27th position, that's a lysine. If you add 3 methyl groups, that'll promote inactivity. If you go over to lysine 36, 2 or 3 methyl groups there will promote activity. And so it's just kind of there are so many different possibilities of what can happen. Again, I don't want you to get lost in the weeds of trying to memorize what these different modifications lead to, but rather think about the time scale, think about the enzymes that mediate these activities and how many different possible methyl groups can be added to a single amino acid. So when we talk about the histone code, it is the entire histone, the catalog of histone modifications that happens to a nucleosome or an entire chromosome. The combinatorial possibilities are incredibly large. And so when we talk about this, this can be happening at the histone level. We can be looking at this and considering this at the nucleosome level. We can be looking down at the specific DNA level with DNA methylation, or looking at higher order DNA in a secondary structure kind of way. The potential here is astronomical. The ability to regulate gene expression in a really complex way is really, really fascinating, but also incredibly hard to study. The ways in which we can study this are still growing. There are really unique techniques that we can leverage to study this, but there's always new techniques coming out to help us interrogate this a little further. And if we took a moment to look at a single histone to see what possible number of combinations could happen, we have along all of these amino acids on H2A, H2B, H3, H4. If you look, each one of those little boxes or shapes is a different modification that can happen at each of those amino acids. So that means at each single histone, there are incredible numbers of, there's an incredibly high number of possible modifications that can happen on just one histone on one nucleosome. There are, I believe, 30 million nucleosomes on a single chromosome. And so this like gets really complicated really fast. But so the key take home here that I want you to note on this slide is that the possibilities are endless here nearly. And it's an entirely new area of active, it's an entirely large area of active research going on right now. Before I jump to our next area of elaborations, I want to pause and give space for a couple of questions on epigenetics. I don't think there have only been a few questions so far, but I want to give space for a couple now. Mm-hmm. When I was talking about the kinetics, that's how long it takes for that modification to happen. Yeah. It sounds like, so to make sure I'm following your question, it sounds like you're saying for the parent cells, it sounds like there's two copies of the chromosome in the primordial germ cell. When we look at the gametes, there's only one copy in each cell. Is that you're asking?
Yeah, and that's going to like the zygote.
Yes.
So they're just not.
Yeah. So our primordial germ cells, they're the like precursor for our gametes, for a sperm and egg. They have two copies of every gene, just like every other cell in our body. But our gametes, our sperm and eggs, only have one copy of each chromosome. They have half the number of that we normally have in our body. And what that means is that when the sperm and egg come together, they each bring one copy of genes, so that then the fertilized embryo will have two copies, as it should. Again, we're going to talk about Klinefelter syndrome and Down syndrome, where there's aneuploidy and extra copy, but typically there should be each gamete provides a copy, and the genomic imprinting are these methylation events at the gametes where they have one copy of each, by nature of either being a sperm or an egg. Does that help clarify? Great, thank you for asking that. All right, I'm going to keep moving. I think we can finish most of it. I will promise not to fly through it. If someone wants, they can make a signal to say like slow down, if that helps. I will not be offended by that. So the first set of elaborations we talked about were epigenetics. The next class and the final class of elaborations are going to be on non-coding RNAs that can actually act at both the transcriptional or translational level. Just to help reorient ourselves, non-coding RNAs they're RNA, so that means they've been transcribed. It's transcribed RNA, or it's transcribed regions of our genome that are not translated into protein. They do not encode for a protein, hence the name non-coding. We talked a lot about during transcription and translation mRNA, and it's easy to walk away from the previous unit thinking at the RNA level, mRNAs are the center of the world. They are not. If we look at RNA in our cells by mass, they make up very little. Ribosomal RNA carries the brunt of the load there. If we look at RNA by number of molecules at the cellular level, mRNA makes up an even smaller compartment of our RNA, where transfer RNA makes up a large number of the molecules there. And so there are a lot more RNA to consider beyond just mRNA when we're working at that level. And so there's going to be other flavors of RNA that we're going to talk about, or at least we're going to try and cover really briefly to try and set the stage for talking about non-coding RNAs. The two that we're going to really focus on are long non-coding RNAs and microRNAs. So two functional meaning that they have a biological function or biological purpose. Two functional but non-coding RNAs that are transcribed but don't encode for a protein that we've already talked about are ribosomal RNA and transfer RNA. Again, they're both transcribed, they're RNA, so by nature of existing, they've been transcribed. They're not transcribed by RNA Pol 2, they're transcribed by RNA Pol 1 and Pol 3. And so we're already intimately familiar with what their roles are. MicroRNA, or you can also call them mirs, are a really unique flavor of non-coding RNA. By nature of the term micro, they're really small, they're on the order of about 20 nucleotides. What they do is they facilitate a really sequence specific, so they complement a specific mRNA target. They facilitate sequence specific or complementary downregulation of mRNA transcripts by a really important process called RNA interference. RNA interference at a big level is a process by which mRNA can be targeted for degradation to lower expression of a gene at the transcript level. This entire process of RNA interference, we're going to go over it, hopefully, today. The entire process won the Nobel Prize about 20 years ago. So it's a really key and fundamental process to biology. Another flavor of functional non-coding RNA that we'll talk about again is long non-coding RNAs. So microRNAs are about 20 nucleotides long. Long non-coding RNAs are, by rule of thumb, about 200 or more nucleotides long. And they act as an RNA sponge, meaning that they will take up RNA by base pair hybridization. And they can also play a unique protein-like function that can actually, by nature of their unique structure, and affect transcription. That's going to be a key thing that we talk about for Klinefelter syndrome today. Two other flavors that we've talked about, or at least alluded to, is SNOR RNA, which is a part of the spliceosome, which makes up the SNRP. Sorry, not SNOR RNA, snRNA. SNOR RNA makes up a unique protein complex that facilitates modifying other RNA molecules. So the RNA interference that we talked about that is mediated by microRNAs, what ends up happening is that our microRNAs are synthesized and they're in their immature state where they have a cap and they have a cap in this hairpin loop on the end. And again, the main part to focus on is about 20 nucleotides long. There's going to be three key molecules that I want to draw your attention to that help promote the maturity of microRNA. The first is DROSHA. DROSHA will clip off the ends with where the cap is here in the poly A tail. And then from there, there's going to be another molecule that acts after DROSHA acts, it'll transport this microRNA out of the nucleus into the cytoplasm. That's where it'll interact with dicer. Dicer will cut the loop off of this microRNA. And once that happens, we have this microRNA duplex that's mature. So historically, we've always thought that with microRNAs, there's going to be two strands here. We've always talked about them as one of those strands really having an effect. So we called that one the guide strand, and we'd say the other one was just along for the ride, and we called it the passenger strand. We've now come to understand that both of them will target mRNA transcripts for degradation. So now we talk about them as either the 5P or 3P arm. So one of these arms will be loaded into a protein called argonaute. Argonaute with one of those strands from the microRNA that's been processed is going to create something that's called the RNA-induced silencing complex, also known as RISC. Once we have the formation of RISC with argonaute and this strand from the microRNA, it's going to be able to identify and target mRNA transcripts that the microRNA is complementary for. And once it identifies those complementary mRNA transcripts, it'll then promote their degradation. And that'll eventually lead to suppressing any translation that might occur from that mRNA. One of the ways we've leveraged this is by synthesizing molecules that will leverage this RNA interference process to selectively silence gene expression in the lab. So there's two methods by which this happens. One is a little more temporary and the other is a little more stable. The first is going to be called silencing RNA or siRNA, where we have a double-stranded RNA oligo or a set of nucleotides that are about the same length of microRNAs, about 20 nucleotides long. It's going to be processed where one of those strands is going to be loaded into argonaute and target an mRNA of interest, a certain gene of interest that we want to silence. And so we can buy these or have a certain combination of silencing RNAs to knock down a combination of genes if we want, but this is one way in which we can silence gene expression in the lab. Another way is with something that's called a short hairpin RNA. What ends up happening is that we have a DNA plasmid or a viral vector here that actually leads to the expression of a short hairpin RNA. And once it's been processed where Dicer cuts off this hairpin loop, we're left with a double-stranded RNA that gets us back up to here where siRNA started off. The main difference is with siRNAs, we're delivering these nucleotides, whereas with short hairpin RNA, you're providing a DNA plasmid that's going to lead to the constant expression of the short hairpin RNA. So long non-coding RNAs, again, they're about typically about larger than about 200 nucleotides on average. They're going to be transcribed by RNA pol 2, 5' capped, spliced, and have a poly A tail. But again, they do not encode for a protein that's translated. There's a lot of functions that they can provide here. They provide a regulation of protein activity. By nature of their large secondary structure, what that looks like is there's all these different domains that can exist within a specific single long non-coding RNA that regulates protein activity. There's also the ability to regulate post-transcriptional control by competing with complementary microRNAs or with mRNAs themselves. And finally, they can also provide transcriptional control. They can actually regulate the entire process of transcription itself at a specific location, and this is going to be key for when we get to Klinefelter syndrome. I think I'm going to go ahead and stop there for the lecture so we have enough time to go over the journal club. So give me a moment to make a note of where we left off. And I'm going to stop the recording here.